What you see in front of you is a picture of plasma membrane. Remember, this is also known as cell membrane. I want you to notice that it is made up of two layers of phospholipids. And keep in mind, each one of these little balls is the phosphate part of a phospholipid, and it has two tails. Also notice that the tails are oriented towards each other, and this inside area of the phospholipid bilayer, notice it's two layers. This inside area is hydrophobic. It really does not like water. Those tails don't like water, so they orient themselves towards each other in order to stay away from as much water as possible. The heads on both sides, they are hydrophilic. They actually really do like water. So if you've got maybe the outside of the cell right here, it's exposed to water, and here you've got the inside of the cell, it is also exposed to water. You have a diagram over here that kind of gives you an image of where this cell membrane, where this is a cross section from. So you can see that this bottom part really is actually facing towards the inside of the cell, and that's why you've got all these cytoskeleton fibers. So that should tell you immediately that this is likely an animal cell. And then you've got the outside part facing towards the outside of the cell, and it's probably going to be butted up against another cell. We also talked about the plasma membrane looking like a fluid mosaic model. So the reason it's called a mosaic is because it has all of these different parts. It's not only made up of the phospholipids, it also has these big pink blobs, which are proteins, and they could have different purposes, but the main one that we're going to talk about is transport. They help transport things into and out of the cell. You'll also see these carbohydrate chains. We didn't talk about them too much in class. They actually assist in identifying different types of cells. Different cells have different types of carbohydrates. And, cells communicate with one another. And then this little gold part, here you have the label, this is cholesterol. Cholesterol is inserted throughout the entire plasma membrane, and its function is to increase the flexibility of the plasma membrane. And the flexibility is where that fluid part comes in, that fluid mosaic model. So you do need to be able to explain what the fluid mosaic model means. Now we're going to be talking about cell transport, and when we do talk about that, we're talking about moving materials from the outside of the cell in or from inside the cell out. When we're talking about transport, one of the things that we need to take into consideration is different concentrations. And when we're talking about concentration, we're talking about how much of a substance there is in a location. So the diagram that you're looking at in front of you now, you have a spread, essentially. On the right side, you have a very high concentration of molecules. There's a lot of molecules within this small area. And as you go to the left, you have a smaller and smaller concentration. Very few molecules on this left side. And it's gradual. Notice it's not just all of them are here and none are here. It's a gradual difference. And this is called a gradient. So when you have a concentration gradient, it means that you have a difference in concentrations from one area to another. So here you have a very low concentration. Here you have a very high concentration. Anytime you are going from an area of high concentration to an area of low concentration, it is called passive transport. Passive meaning that it does not require energy. So every single type of passive transport does not require energy, and it's going from high to low. Also known as it's going down the concentration gradient or with the concentration gradient. Okay? So I want you to notice that in this diagram there's actually three different types of passive transport shown. The first type is diffusion. Diffusion is really the most simple type of passive transport in which molecules, some kind of substance, is just able to move across the cell membrane or the plasma membrane. These are probably very small. They are probably nonpolar so that they can just move right on through. And again, remember that they are going from a high concentration to a low concentration. Here, I did say this was three different types of passive transport shown, but actually we're looking at two. This is facilitated diffusion. Anytime you have facilitated diffusion, it's diffusion still, meaning it's going from a high concentration to a low concentration, but it needs some kind of help. So these purple things that you see embedded in the cell membrane are proteins, and they are carrier proteins. This one in particular is a channel protein, so it effectively just creates this little hole that enables materials, some kind of particles, to go from that high concentration to low concentration across the membrane. And maybe they're just too large to make it through themselves. Maybe they are polar and they can't go through. It can be for multiple reasons, but in this case, all we know is that it's still passive transport because it's going from high concentration to low concentration. The same thing is true here. This protein, it is able to change shape or change orientation. So what happens is that it opens up on one end, allowing one of these particles to come in, and then it opens up on the other end, allowing it to come out. It's still going from an area of high concentration to an area of low concentration. You simply cannot forget that particular detail. That is passive transport. Again, none of these require any input of energy because they are going down the concentration gradient or with the concentration gradient. Now, there is another type of passive transport, and it is called osmosis. You don't see a diagram of this right now because we're going to look at it in more detail, but I did want to just mention that osmosis is diffusion, but it's specifically diffusion of water and water only. So in a little bit we'll look at what that entails when we talk about osmosis. Now let's compare passive transport, in which you have materials moving from a high concentration to a low concentration, with active transport. Active transport, materials are actually being pumped in from an area of low concentration, as you can see here, to an area of high concentration. So the fact that it's having to be pumped means that it requires energy, and that's what this ATP is right here. ATP is the energy molecule, which we'll talk about more later on in the semester. But you do just really need to understand that anytime you see active transport, it requires an input of energy because those materials are being pumped or pushed against the concentration gradient. That is the major difference between passive transport and active transport. You need to be able to articulate that difference and explain, well, if you're going up or against the concentration gradient, it's really almost as if you're going up a hill. So it requires more energy than if you're going down the concentration gradient or going down the hill. What you're looking at now is two pictures. These are both bulk transport, so they both qualify as active transport. They require energy. But there's a major difference in that these materials are actually not going through the membrane. So they're actually getting collected in this little pocket. And this little pocket is actually called a vesicle. And as this pocket enlarges and then kind of pinches off, notice that materials that were outside of the cell are now inside the cell, except now they're inside this little bubble inside the cell. So that bubble is going to go and get processed, and so whatever just came into the cell is going to be able to do whatever it needs to do, but first that bubble needs to get processed. Remember, that bubble is a vesicle. That is called endocytosis. Endo meaning into, cyto meaning cell. So things are coming into the cell. Exocytosis is the opposite. Here you've got some kind of vesicle. Maybe it has some waste products that need to be released out of the cell. And what's going to happen is it's going to come to the surface of the cell and fuse with the cell membrane. When it fuses, it opens up so that the contents of that vesicle can now be released to the outside of the cell. Again, notice the materials are not going through the plasma membrane, but they are getting transported into or out of the cell. So it's still considered cell transport. All right, in the next video, we're going to talk more about solution concentrations and how that has to do with water and osmosis.